All right, good morning, everyone. Thank you. We had a few technical difficulties. My name is Sarah Kostick. I am one of the members of the Plant Breeding Center Seminar Committee, and it is my pleasure to introduce today our speaker today, Diego. Diego holds a Bachelor of Science in Statistics, a Master of Science in Statistics, and a PhD in Statistics. There's a theme there, Diego. He's currently an assistant professor in the Department of Agronomy at the University of Florida, where he's going to be talking about his work in AI and models and all things to help plant breeders become more effective in their programs. Diego, take it away.
Thank you. Before I start, good morning, everyone. Again, thank you for the invite. It's an honor for me to be here. This is my fourth time at University of Minnesota, so first time I'm giving a talk. It's a pleasure for me, and I'll try to share a few of the ideas that we're working on lab of how we understand this idea of conducting predictions and why we want to make sense of how the things that we are doing. So please feel free to ask me any question anytime you want. So I would be more than happy trying to answer the questions. So I titled this talk Multi-omic integration in predictive breeding, hopes and obstacles. And the idea is try to understand a little bit what are the main goals that we are trying to solve, especially if you are breeding, right? Many times we are trying to improve prediction method, but probably the goal is not clear for us what we want to predict. So hopefully this talk is going to help a little bit to clarify those things. So this is the outline. First, I'm going to talk about the context, why we are doing this, and what are the tools that we are using. And then some of the hopes, what are those tools that we can integrate to improve our ability to predict our performance. And then some of the obstacles, why sometimes things might be a little obscure or difficult. And then finally, some solutions, some things that we have developed over time to help the breeders labor. So let's go forward with the motivation. As we know, many of the important economical traits are complex, and these are complex in the sense they are affected by a very large number of small gene effects. What's more important, these gene effects are modulated by the environmental stimuli that they receive in fields in a way that genes and environmental factors interact in new complex ways. So why are we talking about these things? Because as breeders, many times you're interested in improved variety. So you have to observe those things in fields, and sometimes the environmental conditions might be good or might be a little difficult or challenging. So it's gonna pretty much shape the phenotype that you are observing. So for example, nowadays we know that we have technologies like those or methodologies were introduced, for example, by Dr. Bernardo many years ago, where the idea was to predict the performance of new genotypes using genomic or molecular information, right? Back then, the challenge was not to have many genomic covariates, right? They have very few, and still the methodology worked well. Over the time, those challenges changed, and then the problem was to deal with many genomic covariates when we cite these works from McWeezen, where they were futurists and they tried to solve the problem of what's happening when you have too many variables. They invented these improved methods. Well, in this case, we know the phenotypic cost is increasing and sequencing costs are decreasing. The idea is to try to calibrate prediction models using some of the existing data and predict the performance of the observed genotypes using their market profiles. Right? And the concept is very easy. You can just say, I'm going to calibrate the model, I'm going to predict the genotypes, and I'm done. Right? You can you provide those results to the breeders. In theory, the breeders should be open to accept those and use these results to improve the way how they are selecting the genotypes. But we know that in the reality, this is way more complex. Like I said, the concept is very easy to understand, but actually it's very difficult to implement, especially because the breeders are interested in developing genotypes that perform elite varieties in a wide range of environmental conditions. It means that they have to test many genotypes in many different environmental conditions. It means that many times, right, it might happen that the performance of the genotype in one environment is going to be low, and probably in other environments it's going to be higher than the other, right? So this is a graphical representation with only 3 genotypes, where actually one genotype is doing good in one environmental condition and is doing bad in the other. And you have the opposite behavior for the other genotype. And you have a genotype that is stable, right? But it's never the best. So in this case, it's very difficult to pick the best one, the best one. So we have this kind of behavior. We say the very impressible genotype by human interaction. And we might have 3 different ways to deal with it. We can ignore and we can use select, right, for adapted genotypes. The main problem is that we know the environmental conditions are going to change from one year to another, even in the same location. And then we start thinking, what is an environment? A location, an environment, a location, a year, a location by year by planting date. So you plant the same genotypes with a difference of 2 weeks, you will have a different behavior, sure. Anyway, we can reduce it, and we can just take the average means across all of the environments, but still probably we are not getting the best genotypes. And finally, we can exploit. Exploit the sense that actually we cannot test everything everywhere, but at least we can have a very good idea of borrowing information, like between genotypes that were tested in certain environments, but not in others. And this is the way how we exploit. So at least we can have a very good idea of how this genotype will behave in different environmental conditions. And this is good because we can increase, right, the testing capacity. At least we have more hopes or more chances to find the good genotype. And this is a graphical representation of how this conventional selection method works at the industrial scale, where you have beginning a very large number of genotypes you have tested in very few environments, and later on you start selecting the top ones, and then you increase the number of environments that you start this. At the end, after 6, 7 cycles of selection, you end up with very few genotypes, right? They are being tested in many environments. So the idea is that at the end of the process, the company is going to select 2, 3 top performing genotypes, and this will be released as commercial varieties. Now, the important part here is the genetics from the top to the bottom is exactly the same. If we were able to find a method to identify of the 150,000 genotypes, rather 2, 3 that will perform everything, we can save a lot of resources like land, water, seed availability, but more important, time, right? Obviously, it sounds impossible, but the idea is trying to get somewhere in between, right? To short the selection time. If we analyze a little bit more this diagram, we have that the total number of genotypes or phenotypes that we need to screen everything in one step is 150,000 times 100 environments is like, yes.
Okay, very nice.
Oh, okay. Yeah, it's 50 million combinations, right? We know that we don't have enough seed, but that will be the ideal. Now, if we analyze the whole pipeline, we have around 12% of all of these 50 million combinations are observed in the fields, assuming that the 100 environments we have there encapsulate the initial 10, 20, etc., etc. So they are already spending resources for observing 12% of all of the combinations. So probably they can work with some sort of sparse testing design in a way that should be able to evaluate most of the genotypes somewhere in the field. And then here is where we start thinking, what would be a good method to screen in or to screen out the good or the bad genotypes? And sometimes we start thinking whether multi-omic integration can help with that, right? We have not just genetics, we can have phenotyping, weather data, etc., etc. And I think that the message is it is going to depend on what's the final objective, whether we want to predict phenotypes or we want to predict breeding values. As far as I know, breeders are more interested in predicting breeding values rather than phenotypes. I don't know if that's the case, but this what I understood collaborating with colleagues. So let's see a little bit more about this. Is when we start thinking about what we call the omics bets, and it sounds like a superhero movie, right? And this is where we have some of the hubs. So what will be the different layers of information that can help with this? We know, right, the plant performance is affected by many, many different factors. One of the most important is genetics, but it's not the only one. And the idea is to integrate as much information that we can collect around the breeding process. So this is a graphical representation of one of my postdocs. We have to put together all of the current different omics that we can work with, besides genomics, transcriptomics, proteomics, phenomics, enviromics, and some of these have advantages and disadvantages. And other omics like ionomics, right? The amount of the specific elements in the tissue. So all of these things can be included in the prediction framework, but it's important to try to distinguish which one will be more useful or which one is going to bring more information. So some of the terminology that we have is, for example, we are using genomic information. We have something that we call genomic selection, and it has many advantages, right? I think the main or the principal advantage is that we can have an idea of what will be the performance of the genotype without serving fields, right? We can just skip at least 1 year the breeding process. Disadvantage is that many times it depends on predictability, and more important, the environmental stimuli, right? Sometimes we have traits that have low performance, so probably the genomic information is not perfect, etc., etc. So it's good, but sometimes we have some gaps there. Phenomics is a good one. It's one of my favorites, right? You can increase predictability. You can usually outperform genomic information, and combining with genomic information, you can have a little boost in predictability. What's the main problem? The problem is that you have to observe these in fields, right? You have to collect, you have to plan, you have to find a way to justify value using genomics. And one day I was giving a talk in economics using prediction, and somebody asked me a question. Yeah, what's the advantage? I mean, you can just wait a few weeks and then you can use harvest and you have the phenotype. You say, yeah, you're right. So we have to justify probably with a trait is very difficult to measure, right? Like making time of this quality traits might be very difficult, very expensive to measure. You can say, oh, I'm using economics for this. Other than that, it's going to be a little difficult to justify values. Enviromics. So the advantage is that we can characterize difference between environments using weather data, and the weather data is public available. You can find it much everywhere from NASA Power, many repositories. However, one of the disadvantages is the probably the set of environmental covariates that we can integrate in the models is not enough to fully characterize the difference or similarities between pet environments, but still it's not totally complete information that we can get from there. So these are just some examples. Now, regarding the prediction framework, okay, we have our favorite methods, statistical models. Why? Because this is a oversimplistic representation of the reality, right? We are trying to address or to explain everything about the plant performance using a set of parameters, and we are kind of forcing the parameters to explain some of the variability. Easy to interpret, right? Sometimes might be a little obscure how we implement this, but still the easiest to work with. Right, every parameter has to explain something, right? We address directly a specific percentage of variability. Second set of methods, non-parametrics for AI, right? Now everybody is talking about AI, ChatGPT, etc., llama models, etc. These are good, but they are some sort of black box type, and they rely many times on hyperparameters. You have to optimize what will be the election of hyperparameters to make it work very well with your data set. Number of hidden layers, optimization function, dropout, learning rate, etc. The disadvantage is that many times the machine could trick the user, right? The big idea working with these AI methods is that you have to minimize a loss function. So the difference between the square of the difference between predicted and observed values. And many times this machine is going to pick the layer that minimize that difference. And that difference might not be even genetic, it might be weather, it might be management. And the readers might be very happy that you have a method that performs very well trying to predict, but probably you're not advancing genetic, you're advancing something else. That's why it's a little tricky when we think about using AI, right? And finally, cross-broad model that relies on constants, right? The information that they deliver is important, right? We can learn a little more about development of the genotype, development stages, yield components. However, getting this constant might be a little complicated, expensive. You need a very comprehensive experiment to start learning a little bit more about this constant, whether you set the CN, et cetera, et cetera. So these are kind of some of the methodologies that we can implement. So let's talk a little bit about the obstacles. And this is the important part. Many times we might have some problems trying to interpret the results. Again, the goal is not predict the performance. Probably for a breeder is to advance the debate. So adding all this data in a good function can help to improve predictability. However, it might be a little difficult to try to address what is the specific or pure signal that one of these components is explaining, right? So just imagine a little bit, when you go to the supermarket and you pay, you get the receipt, you know pretty much how much you pay for each one of the different items that you bought. Now just imagine that you lost the receipt. And the only thing that you know is how much you pay, and then you start trying to guess how much each one of the different items costs. And now actually you have a very good idea, right? Like, okay, milk probably not that much expensive, eggs, or you bought something that's very expensive that you can kind of rank. But still you have uncertainty, and that is the things that were happening in our case. We don't have all of the set of variables, or the set of variables that we are using does not explain every single process that's happening in the field. So that's why it's a little obscure. In the absence of one of these variables, right? The variables that we have left, like when we start fighting to explain that variability, that was not explained, is not explained anymore, but that variable is out. So they start fighting. So again, the problem there is to create this orthogonal set of information that, you know, the genetics is going to explain some specific information, but it's not going to overlap with other layers, right? In the absence of genetics, the other layer is going to start explaining that missing parameter. Still, we haven't mentioned anything about interaction. We know that many factors interact: genes and environmental conditions, phenomics and environmental stimuli, or interaction between omics, right? Like bionomics, for example, the elements in the tissue, genetics, etc. So this gets way more complex. And it's when we start thinking, what do we want to really to predict? The phenotype or the breeding value, and this is when it becomes a little complicated to find an answer, right? And many times when I talk with the students is, then it depends what do you want to predict and what is the stage where you are working in your breeding program. It will matter whether you want to predict one or the other. And this is an example that we work using soybean data. I'm still working with soybean also. Where we try to untangle a little bit the variability. So we have in the top left, the effect of the field variation, where we have row column and blocks. In the top right, we have the estimated breeding value. In the bottom left, we have the noise, right? The information that we were not able to explain. And in the bottom right, the trait. So the sum of the three panels return the yield. And what we find here is that actually we did a very bad job trying to get the breeding value, because I don't know if you can see this part of the genetic effect resembles the phenotype and probably is very related still with the field variability. This is just one example. Still, we carry a lot of the signal is coming from the field variability, and we would expect that whatever we are working there in terms of the genetic will be assigned randomly, but basically patterns that still carries a lot of that thread. So probably we are doing a bad job. And this is working with special adjustments. So it's very difficult to untangle the source variability. The better the estimation of the breeding value should resemble the less the phenotype. Why? Because the phenotype is going to carry all the signal, it's not just genetics. Obviously, we don't have these parameters for comparison. Nobody has the actual breeding values. And then when we start thinking what to do, I should contrast versus what. In any case, predictability is going to reduce. The better you get the breeding value, the less is going to resemble the phenotype. It means that probably your prediction model is going to underperform compared with any conventional method. And then when we start thinking, are we being unfair with the approach? Many times we blame the model. The model is not working well. Actually, you don't have the correct values to contrast whether the model is working well. Again, many of the times we are resemble. In this case, it's just one number using my field variability, but we know that we have other things going on at the same time. It could be weather formation, management, etc., etc. So again, still, right, breeding value resembles the phenotype, and we expect that it should be randomly assigned. Is when we start working in a little bit to conduct the analysis. So in this case, for some reason, I cannot see the first column. I don't know what's. Anyway, let me escape a little bit. No, for some reason, I cannot see the. Sorry, I cannot see the different factors in the first column, but actually there we are trying to compare the performance between yield, phenotypes, and some sort of adjusted values. Right? In the first row, in the first column, sorry, we'll have the correlation between yield, and that correlation is 1, right? After that, we have the correlation between adjusted value of yield versus yield, and that decreases 0.52, and it depends on the model. We have models that include block effects, row column interaction, etc. So the idea is that the more information you put in the model is going to decrease the performance. Right. In the second case, we have 1 second when we contrast with estimated values. In this case, in model 1, we have the estimated value when we use the line effect only, right? And when we compare with the phenotype, is 0.95. When we compare with something that we try to extract from the experiment, the actual breeding value, it decreased to 0.05, 0.051. And when we compare with a model that includes row column, that value decreases. So pretty much the more information we are adding to the model, meaning more data, right, more omics added, the less the estimated value is going to resemble the phenotype, and it's something that we expect, right? Because in theory, there should not be any trend in the model, in the data, how you plan genotypes in the field. And it's what we observe. So many times we have this question, what will be the best way to contrast, right? What will be the best way to evaluate the model? Nobody has the breeding patterns, and this when it becomes a little challenge. So one way we did to go around with this was to conduct a simulation study, right? Where we know the breeding values, we know what are those. And then we simulate, in this case, field variability in the left side. And then we add some block effects. And then the plot in the middle shows, right, the breeding value. And as you can see, that thing is randomly assigned. We don't have a specific pattern. Obviously, we have to add some noise, some variability that we cannot explain. And finally, the phenotype is going to be the sum of all of the previous components. And then in the phenotype, we can see that there is some trend, right, it's related with the field variability. Now, in this case, we can contrast whether the model is doing good or bad, predicting the breeding value, not something that is coming from the phenotype, adjusted from the phenotype. Okay. Again, the better the adjustment is conducted, the less the breeding value is going to resemble the phenotype, and that makes sense because there is no reason to think that those were assigned in specific order in the field. That was randomly assigned. And again, sorry, for some reason we cannot see the characters in the table, but I will try to explain a little bit. So the first column represents, right, the correlation between the, the phenotype of whatever we are using to estimate the breeding value versus the actual breeding value. In the first column, we have the model that includes only block effects, and you can see when we have more complexity, we start improving the predictability. In the second column, we have the contrast between the predicted values or the adjusted values, right? Using genomics. And again, the more the model is being more complex, the best. We keep increasing predictability. In the last column, we have the contrast between the actual breeding value and whatever we are predicting with the adjusted values. And as you can see there, the predictability pause. So pretty much in the real life, we will have pretty much the results from the first column because we don't have the actual breeding value. But if we were able to have the actual breeding value, the results of the model will be way better. And I have been talking with people in the private industry, and they say, yes, you are right. Many times the predictions are doing way better than whatever they are estimating using phenotypes. And this is one of the reasons, right? We are trying to untangle better that signal, but we don't have the actual values to compare with the model is doing good or what. At least we can try to get these answers using simulations. And here is where we start thinking, so what's the ultimate goal? Right? If a breeder wants to increase genetic gains, actually the breeder should be more interested in being able to estimate that breeding value. Again, the best is to estimate or to isolate, right, this signal, and then select based on this estimated breeding value. Probably a private company, after they develop the improved genetics, they want to make contracts next year how much seed they can deliver. So they have to estimate with high accuracy how much product they will have. And it's when they are more interested in predicting the actual phenotype. And it doesn't matter if whatever is driving the variability is something that they can control or not, right? Like whether so they already developed the improved genetic. They just want to know how much they can compromise to sell in a contract. So that's kind of the idea of the different approaches. Right? So as breeders, we might be more interested in the first one. Private company that wants to make a business with somebody else might be more interested in the second. It doesn't matter what is the omic is target the variability. So some reason it's not working, but in this plot, whatever we have in the inner square corresponds to the actual breeding value. At the top, we have the correspondence between the actual breeding value and the phenotypes. And you can see there you have many crosses, right? The correlation between the actual breeding value and the phenotype is 0.65. In the bottom is the correspondence between the breeding value and the field variability. And as I said before, there is no reason to think there is a specific correspondence. So these are assigned randomly. Correlation is practically zero. In the left side, in blue, we have the correspondence between the breeding value and whatever we are estimating, right? As part of the genetic signal, and that goes to 0.81. And finally, in the right side, we have the correspondence between the breeding value and the predicted breeding value. And that case goes to 0.944. So that's interesting because actually the model is not that bad. The model is bad when you are contrasted with something that is derived directly from the phenotype. So probably the model is doing a better job than what we expect. We just don't have the parameters to contrast the performance. Okay, now I'm going to switch gears. I'm going to talk a little bit about some of the solutions that we have been working in our lab. So first, I'm going to talk about Chido. That's a word in Spanish from Mexico, I guess. It means cool. So in this case, Chido is a no-code application that aims to remove these technical and financial barriers associated with the model of interacts. Pretty much how we can implement these things in a scale where we have limited resources, right? In the prediction context. It stands for characterization and integration of driving omics. And this software was initially developed by couple of students. So Francisco Gonzalez, he was a student with me. During the day, he was a student. At night, he was a model lead, so Google North America. So I thought he might know a little bit about programming, and then we took advantage of skills. Second one was William Garcia Vadillo, was ex-intern at Google. He was helping a lot to developing the initial phase and still keep developing. And finally, we have Victor Segues, not a visiting scholar from Brazil, now is breeder back in Brazil, working with soybean. So he took Chido to the next stage, right? How we can use large data sets, talking about millions of data points, you can conduct analysis using GPUs. And the idea is very simple, right? Because the motivation started when I was consultant for CIMIT, I guess, right? I used to get a lot of databases. I have to run analysis, and many times something was wrong with the database, and I have very long codes, and I have to modify everything in the codes. So I thought we have to have a standard how we are going to provide the data, and then after a specific point, it should be straightforward how we implement analysis. And the idea is create sort of covariance structure with whatever data that we can provide. In this case, if we have genomic information, right, at the line level, we can end up with some sort of covariance structure, right, the result of the similarity between person individuals. We might have another sort of information that is probably specific to the plant or the plot level, right, where we have a lot of GY coming in, and we can develop another kernel. So the idea is to multiply, right? First, we expand the covariance structures, and we multiply this to create some sort of interactions. And this is the way how we can actually model interactions with every single factor in one layer and every single factor in the other layer. In a nutshell, this software checks data integrity, connects omic data sets, it uses specific columns, right, to associate phenotype with the different omics, generate some of the cross validations that we might be interested, collect the statistics to evaluate the performance of the models. And currently, we can conduct forecasts using general combinability, hybrid prediction, special combinability, host pathogen interaction, stability studies, and the next releases will include the analysis of GYE, sparse testing, and deep learning. So some are kind of the next stages that we are trying to target soon. So how can I have access to this? You can work on demand. So we have a server that you pay, you can just go, pull up your data and play with the implementation. It's like a video. Can you develop the models like drag and drop interaction, drag and drop different terms? Or you can download implementation in your computer. So in that case, right, the limit is the power that you have in your computer. In the future development, we are going to try to post this in the hypergator, the supercomputer we have in UF, where we can use take advantage of the GPUs to run analysis with very large data sets. Another implementation that we developed to deal with multi-omic integration, in this case was for categorical data, was something that we call one-pass method. It's a collaboration that we have with statisticians from different universities. And the idea was to develop a classification method that can outperform conventional parametric and non-parametric models. And the idea of one-pass method is very easy. So you can theorize what is the data set that you start analyzing first. When you combine molecular marker information, you can have like millions of markers. Secondary traits, you can have 20. And you treat them as the same nature, right? The information that you have in the secondary traits is going to disappear. It's going to vanish because you have too many markers that are going to start fighting for explaining that portion of variability. So the idea behind this development is we start selecting, right, the variables that explain variability the most first with the layer that has the less number of variables, and then we start selecting conditional on that from the next set of variables. And you can put together 6, 7 different omics, it's going to work well. So there we try to work with a classification. So we have binary case, yes or no, present or absent. And we evaluate the model's performance based on the classification error. So the lowest the classification error, the best. In the left side, we have the performance that we obtain using the AI methods, both FTA, random forest, or vector machine. In the right side, we have the result that we obtained using the method that we are proposing. As you can see here, most of the time, right, the method that we propose return a reduced classification error compared with the different machine learning approaches. So the idea is take advantage and be able to explain why we are getting the results that we are obtaining, right? At least with the method that we propose, we end up with a very reduced set of variables to explain that variability. In the other cases, we needed around 2,000-3,000 different variables to explain that trait variability. So it has some advantage. Another method that we developed has to do with combining outputs of multiple prediction frameworks. This is a study was developed for a couple of scholars from Brazil that we have last year, Dr. Moisés Nascimento and Ana Carolina. They came to Florida and they had this idea of combined outputs of different methods to take the most advantage of the best of the best of each one of the different methods. So you block, we have infinitesimal model, quantile regression, you rely on quantiles, how good or how bad you are able to explain those differences. Multivariate adaptive regression, that's an extension of linear models that automatically start modeling nonlinear relationships, and quantile random forest, the combined random forest and quantile regression. So the main goal was to combine or find a method to combine these different base learners. And what they found is actually when you select those base learners that perform above the average of all of them, you can improve predictability. In this case, they implement this in Copia Arabica. Predicting yield, they found that this method outperforms U-block by 87%. Predicting total number of fruits, 37%, leaf mean infestation, almost 200%, and second scoliosis, 15%. So they found a method that how they can integrate, right, the different abilities of the different models to improve predictability, rather than combine all of them to treat them as the same network. Okay, now I'm going to talk a little bit about using AI methods, and this is not for criticizing the AI method, but at least is for have an idea how we can take advantage. So we live now in a revolution of AI, and everything is AI, and I don't know how many of you have used ChatGPT and all of these llama models to improve performance. At least in the business we are doing in terms of predictability, when you compare all of the methods that have been published, you find that there are not actually significant difference, statistical difference between the new methods and the conventional methods. However, those new methods had advantage in many of the libraries were written to speed up the analysis. So I have a student that thought, why don't we take advantage of these libraries that were developed for AI methods and we implement those using conventional parametric models. And he started talking to me, okay, we have a problem, right? We have depending on the scale of the problem, right? We need specific GPUs or CPUs to deal with that. We have too many data points. It's way easier or more convenient to use a GPU than a CPU. Again, the idea was to use all these advanced developments to take advantage in the implementation of conventional methods like G-block, random, R-block, RKH, Bayesian alphabet pretty much, and how we can leverage those things for analysis. Again, if the advantages in predictability are not significant, let's continue working some of the methods that we already developed. Why? Because it's easy to explain, right? The variability can be explained or addressed for different components. Is when this student, Victor Seguet, now in Brazil, said that he will use Python, so he will translate everything we develop in R to Python and use these libraries from Rapids, QDF, and CuPy because those are very good to handle large data sets. So he combined this with the hypergator, the supercomputer. We have a UF where we have this brand new GPU processors. And then he started challenging the implementation with some real data. So he took a data set that we have a large data set of 2,500 genotypes tested in 340 environments, soybean data. All the genotypes tested in all the environments for a total of 850,000 data points. When I started working with Dr. Lawrence many years ago with soybean, for me it was a dream trying to model all of the data that we have in soy now, about like 60,000 data points. I was never able to even develop the incidence methods. So this was like my wildest dream to try to analyze this data set, believe me. So he put together everything and used two cores AMD processor, 128 gigabytes of memory, and back then he used the A100 NVIDIA GPU. Now we have B200, and these are the results that he obtained. So I started asking him, why don't you start from the easy, right? Like, let's say you analyze 100,000 data points and see what happened. Now 200, 400, 800. And then he found that actually when you double the size of the phenotypes, the time to run the analysis is not increased it's not double the time. Actually, it has a slope of 1.7, so you can keep increasing, but actually the time is not double the actual time from the previous set. So with 100,000 phenotypes, it takes like 6 minutes to run a model with many effects and interactions, all the way to 850,000, where it took around 40-45 minutes. I think sometimes we can wait, right? Like obviously not going to be like 2 seconds. But still now we can take advantage of all of the different data points that we have in the models. This is how we tested. So it says, let's go and predict the performance of 500 new genotypes of the 2,500 across the environment. 340, and let's see whether the conventional model based on main effects is doing good compared with the interaction model. Actually, he found the interaction method was always better, or most of the time better than main effects. It was just for us to check that the model was working well, the implementation was working well, they keep sampled, etc., etc. Again, we can use or take advantage of the use of GPUs for fitting genomic prediction methods, specifically using parametric approach. Why? Because we can address, or it's more tangible, what is the contribution of the different components in the model to explain the variability. And in the long term, we will see this GPU programming is evolving very fast. So now it's just a technical issue. It's not something that is going to prohibit you to use large data sets in the future. Or we can wait a couple more years and we'll have another more powerful GPU. When Victor worked this with the new B200, I think from 1 hour you reduce to 15 minutes. So you can imagine that the more these things are going to be evolving, the easier for us going to be. We will be able to run more large data sets. And finally, I'm going to talk about the use of genetic diversity as a proxy to select outperforming genotypes. We know with genomic selection, many times we are trying to use a training set that we observe in the fields to predict the performance of genotypes that we haven't observed, but still we need some tangible information that is observed in the fields. So we took advantage of some method that was developed in this course in the University of Nebraska, supersaturated design. Actually, the paper was published 4 years ago. Dr. Lawrence was the lead author, or corresponding author in that paper. And this method attempts to select the set of genotypes that maximize genetic diversity or minimize genetic diversity. You can play with both ideas, right? Let's say you have a target, you say, of 10,000 genotypes, I can certain fields only 100, which is the one that maximizes genetic diversity. And let's be clear, it's maximizing genetic diversity is not the one that is more representative of the whole population. So these are different things. So we thought that there might be some correspondence between genetic diversity and performance. So we start sorting the genotypes from the most diverse to the less diverse, right? And we found PC1, PC2, the most diverse will be in one side, and the less diverse will be somewhere in the middle. It makes sense because they are more similar to everyone. So cannot be in the other extreme, it should be somewhere in the middle. When we compare with the stability parameters, intercept and slope, we have something like this, a little weird behavior, because we have the most diverse above a specific diagonal and the less diverse in the other diagonal. So we couldn't find a specific clear pattern. When we compare performance, in this case, in the pie-axis, we have the average yield across environments. We see a little bit of a trend, right? In this case, the most diverse have a higher performance compared with the less diverse. And many times that doesn't make sense because we would have expected the opposite, right? The less diverse have elite variety, they have been over-selected many generations, and usually they have a higher performance. In any case, we realize actually we are finding in the extremes the good and the bad ones. So, and this method can help for that. So when we compare with the stability parameters, again, we don't see a clear pattern, but at least we see that those that are more diverse have a higher performance. We can compare with the slope, with the intercept, and this is kind of the key result, right? The vertical dashed line corresponds to the most and the less diverse. In this case, the most diverse have a higher mean. When we pick the extremes, we have the most diverse have a significantly higher performance than the less diverse. At least we have a method to screen out bad genotype, right? We can just focus on those, and one of the advantages is that you have higher genetic diversity, so you can use those for the breeding program to ensure you have genetic gains in the long term. And then we start exploring other ideas. What happens if I already have a core of genotype that will be my target, and I have a specific amount of resources to plan in fields for training the model, what will be the best way to do it? In the left side, we have in orange the target set. In black, an initial random sample. And we use the supersaturated design implementation to start selecting the set of genotypes, the maximal genetic diversity in the middle. So those that are more diverse than the testing set, and the minimized genetic diversity in the right side. So those that are more similar. And we found something different to what we expected. Obviously, in this case, we had to focus in the model. They had the main effects in gray color, because other models have more omics, so more difficult to interpret and to explain. And it's clear that the most you increase the genetic diversity, the performance of the model is the best. So this is an iterative process that you have a random sample, then you start selecting one genotype in, you take one out, and then sequentially you minimize or maximize genetic diversity. So conditional genetic diversity. So we can track down what is the performance of the model as we increase or we include or exclude genotypes. So the dashed line corresponds to the case when we are increasing genetic diversity. The bright line corresponds to when we minimize. And we thought that if we select something this close to the testing set, it's going to work better, but no. The problem is that artificially we are reducing the number of effective phenotypes for the model. We have, let's say, 200. You have randomly selected 200, you have 200 for training the model. In the other end, probably you end up with still 200, but those represent probably 50 genotypes or 50 data points. So you are decreasing because it's very similar to whatever you have in the test. So we found in this case that the best way was to maximize genetic diversity, and it works better. Now, when we look at the blue line where we have GY interaction, similar behavior. We have the orange line, we have family interaction. We don't see any advantage. And again, this is the case when you have multiple omics. The more you complex the model, the less you can interpret, or it's less tangible the result that you are obtaining. With this, I want to end up, and I want to invite you, if next year you don't have anything to do in the summer, we welcome you to North Florida to join the multi-omic integration for AI genomic prediction breed short course that we are organizing. Believe me, you will have a lot of fun. We have a short course where we invite more than 10, 14 different professor, researchers from different places to come and present in a workshop the things they are doing. A lot of practicals, data handling, data coding. So please consider to join. I'm trying to convince Dr. Rex Bernardo to join us next year. We have instructors from multiple universities, research centers, private sectors, and we already have a lot of attendees, over 180, 34 countries, 5 continents, many states in the US. So we already have some folks from Minnesota. With this, I want to thank you again for your time and for your interest, and of course, go Gators. Yeah, I have any questions? More than happy to. Yes, please.
What's the metric you're using for most diverse and least diverse? How are you measuring genetic diversity?
Okay, when we think about a covariance structure, like you get markers, if set is more diverse, you might think that whatever is in the off diagonal will have a correlation close to 0, right? Cannot be very positive, very negative, because you have some trend. Compute that average correlation of the off diagonal is a little complicated. So it's equivalent to compute the trace or the matrix of the diagonal. And it's what we use. So instead of minimize the off diagonal, we maximize the diagonal. If you want to increase genetic diversity, you want the opposite, okay, we minimize that value. So you start with a random sample, and then you test every position in the target set, right, with every position of the universe that you have to choose from. For example, you have 100 genotypes you want to select, and you have 1,000 in the universe. Number of total combinations for one iteration is going to be 100 times 1,000, so 100,000. You select the one that has to go out and the one has to get in. And then iteration 1, and then again, you start repeating another 100,000 combinations to select the next one until you reach conversion. Yes.
Continue with that question. Did you compare your scores with NA genetic diversity?
We compare with FST, and then we found, because we wanted to validate if the method is good for ranking the genotypes in genetic diversity, so we create groups of genotypes. The most diverse, the next group of 40 genotypes, the next group, and they match almost 1-to-1. The only difference was with the less diverse, because that less diverse is similar to everything, even to the most different. But in general, the trend was very clear. Yes.
For phenomic selection, where you use drones to substitute measurements, substitute for markers, I appreciate what you mentioned, that the limitation is, well, you need to roll the plants to take those measurements. Are you aware of any literature where you minimize that? Say, what if I take my drone measurements from only one plant, maybe at the seedling stage? Do you know of any literature along those lines?
I don't know a specific literature, but now we are working in a project with Dr. Whitaker in strawberry using the measures taken at the seedling stage. So they will be planting those in fields sometime in next month. So let's go this forward. Yes.
There's a question online. It says, I was wondering how you're adjusting for spatial effects in your analysis. Are you using a regression-based correction, a mixed model with a spatial covariant structure, or some other approach?
We want to have something very standard, because I know there are very sophisticated methods that use splines and they work well, but the problem is when you want to reproduce, and whoever runs the analysis is going to get something different. So we want to get something very standard that anybody can reproduce. So we use mixed model approach. Yes.
In terms of randomizing your trials, so as you move from, randomization trials for purposes of just cohort selection based on the phenotype of the line per se, towards randomizing trials for training prediction models, have you thought about incorporating the genome-wide marker data to maybe optimally randomize trial to train an optimal model? Because now unit of randomization could be real rather than force your time.
Yeah, but I mean, one of the problems we have there is still we have some trend in the field. Probably the signal is larger than whatever we can capture from genetics or genomics. So we have thought about that, but we are looking for collaborators who can plan those things in field. Because I mean, I'm just a poor statistician. If I go to the field, I don't know what to do. No, I'm being honest.
I have a geeky question. So 0.36 seconds to invert a 100 by 100 matrix. That seems slow. I was wondering what language that was.
Okay, but when I thought about the same, I asked the student because he put together, it's coming from a paper. I'm trying to remember it was an inversion or something like that. I really don't remember but it's in the paper. Sorry, I don't have all the data, but to me was like, okay, I thought they were kind of similar approaches and you get an inverse very quickly nowadays. So I don't know what type of operation was that. Yes.
So they're not missing environmentability. So you mentioned missing heritability, and there is something similar with the environmics. Like that.
Yeah, actually, I prefer to call missing comparability because we don't have the breeding values. So we can simulate those things and we can observe that it is working well. But I don't know if I would say missing heritability or environmentability. I think at least we don't know, we don't have the contrast. And all the time we are trying to force to contrast with something that is derived from the phenotype.
So do you see similar problems, issues with other omics data, like transcriptomics? You mentioned the omic run. The similar problems.
I mean, many of these other omics depends on the environmental stimuli. So you already have that embedded the interactions. So sometimes it's you can improve performance, obviously, but I don't know how easy it is to interpret and translate to another set of environmental conditions. That's the main problem.
Sorry, I have one more question.
So please.
So, because I don't get to interact with AI experts too often, but I've thought, can you train a machine to replace a breeder? I'm sure you've thought of that, but if you think of the tasks the breeder does, can machines be trained to do those things? Because, they're cheaper.
Yeah, but they are also make mistakes. So you cannot blame the machine. They're gonna fire whoever is using the machine. I mean, it's interesting. It's just another tool, right? And there are some collaborations. We're starting now with a private company in France, and the idea is to use all these ideas, embeddings, transformers, to see how much we can improve the predictability. And that would be like a dream someday. Like you have some similar thing to ChatGPT, and you just provide data set, and you say, pick me the best one, right? Like, based on a specific set of criteria. I don't think the breeders will be substituted anytime soon. But what I saw working with people in private companies, they require more skills from the breeders of the field, not just being breeder, quantitative statistician, using phenomics, AI, etc., etc. And the problem is, the more broad you are, the less you can specialize in something. At the end, it's a team effort. Yes.
Once you have that robust set of data, can you give us a call? Can you generate the synthetic data to train the model?
Yeah, it's something that we have done with some private companies or with some big collaborations where actually we get the synthetic data, we predict those things, and we integrate those as part of the training stage. Obviously, there are some things that we have to consider because when you predict something by design, the predicted values are strong. So you have to find a way to return those to the right scale and the correct mean. But yeah, you can increase artificially your training set sizes.
Any final burning questions? Oh, let's thank Diego then.